Welcome back, everybody. Today we're going to talk about sponges. Before I start, I want to let you all know I apologize if I sound congested or if I cough or if I sneeze. I'll try to edit those out. I do not have the coronavirus, or at least I don't believe I have it in any way or form. I have allergies that I often get when I don't sleep well, and that's kind of what it is. No fever, feel fine other than that, but here we go. Okay, so today we're going to talk about sponges, the first group of animals on planet Earth. Okay, so first of all, the sponges have a cellular level of organization. I think we mentioned that already, which means they don't really have tissues. Mostly marine, about 9,000 species. There are some freshwater ones, but they all live in the water. Part of that is because of how they feed. They're a type of like filter feeder, and so you can't really do this on land. So all entirely aquatic. Mostly in the ocean. They are asymmetrical, which means they don't really have a clearly defined body shape. They're kind of like a blob in a sense. And they are sessile, and sessile means they don't move. They are also true hermaphrodites. We'll see this term many times, and we'll talk about what a hermaphrodite is. But essentially, a hermaphrodite is an animal that, or another organism that can reproduce sexually and has both male and female functioning parts on the same organism. So that's the definition of a hermaphrodite. And there are true hermaphrodites in the animal world, including the sponges and many worms. And again, that means they can produce both sperm and both eggs functionally. They usually don't fertilize themselves. I think we mentioned this with plants already. There's mechanisms that prevent that, but that's what a true hermaphrodite is. Now, in the case of humans, there's a wide range of topics about sexuality, which you can take whole courses on that are very interesting. But sometimes, just this is my little tidbit here, some people occasionally accuse or use the term hermaphrodites very inaccurately for certain animals and for people, which are a type of animal in a sense. But the reason that is not accurate is because in many animals, like in a human, the parts that become the sperm producing parts and the parts that become the egg producing parts are the same tissues, and then based on a variety of genetics and hormones and the interactions of all those, humans and other animals develop in one or more pathways, but they don't produce sperm and eggs functionally in the same organism. So anyway, in case that comes up. They often live in groups, and the group of sponges is what we call a sleaze. If we look back, remember we talked about our animal cladogram. Here it is right here with all our different organisms. And just sort of to put this whole thing in perspective now, just so you know, in the very beginning of class, I tried to make the argument, what's really neat about Bio 2 is when you're done, you can go out into the world, and no matter where you are in the world, anywhere on the planet, anything that's alive, you should be able to sort of be able to identify it and put it in one spot or another. So if you, this doesn't have our bacteria and our archaea on it, but if you look here, we have all the supergroups of the organisms we did in the eukarya. Here's our animals right here, right? And so if you just sort of attached this cladogram to that, you would have what you have there. So everything sort of ends up fitting somewhere and has characteristics that put it there, and that's where we're headed. Now, When we talk about sponges, sponges again have a cellular level of organization, and there's a couple sponges that are or cells that are particularly important. First of all, we have our choanocytes, and our choanocytes are a type of flagellated cell. You might remember I showed you something like this when we did the Uniconta, and we talked about these particular cells, and this is where I said they'd come up again. And so the choanocytes are flagellated cells with these little extensions that catch food particles, and they have a flagella. The flagella beats and creates water flow, and it picks up those little particles as they flow through. And then we have these other cells called amoebocytes, which are like amoebas, which have pseudopodia. Here's my amoebocyte. And those essentially can move around throughout the sponge and pick up the nutrients, and that's how it gets a lot of things done, transferring nutrients around. So no circulation system or any of that kind of thing. We're talking about a very simplistic animal that is at a cellular level of organization, and the cells even don't really form a tissue of any sort. They operate somewhat independently. Okay, now in terms of the body or the shape of the sponge, there's a couple of things that lend to that happening. The skeleton. The first is they can have these spicules, and spicules are like little spines. And like we've seen before, spicules can come in two sort of chemical types, calcium carbonate or silica. This is like a glass-like material. And then this is a white, hard material, kind of like what you see in a snail shell. So when we did our supergroups before, we talked about the test of diatoms and dinoflagellates and things like that. And we saw those tests or shells, if you will, being made out of those materials. So that's one. And then they also have this protein, and the protein is called spongin, and that's what you see here. So those sort of give the shape to the sponge. There are three kinds of body shapes that you find in a sponge. One's called the asconoid body type, and that's the most simplistic. And what happens in the asconoid body type is water through those beating flagellated cells goes through these openings called the ostia. It flows from the ostia into this big space here called the spongiceal, and then it goes out the top, which is the osculum. So that's one body type. The second body type is called the synconoid body type. And in that type, I might have spelled this wrong. I keep thinking, have I spelled that wrong, or have I fixed it? And then is it Friday or is it Wednesday? It's confusing times. My kids, by the way, so, just to keep this authentic, because I want it to be just like what you would get in lecture normally. We're in this pandemic and online learning and all these things. And my kids, of course, are home too, like the whole rest of the world. And again, I'm not sick, but so we're doing online learning and all that at home. So we're working from home and we're teaching from home and we're having kids at home. And then next week, Monday, my kids have spring break. I mean, what is that all? What do we do on spring break? It's like exactly like every other day. So I feel like we're getting ripped off a little bit here with this spring break thing. And I'm suspicious about it. Anyway, another problem. But I want you to get the stories. I would in class give you the story. So I want to give it to you. So there you go. Anyway, synconoid body type. Same kind of thing, but more complex. It's a more complex body type. So in this case, we have our opening once again. There's our ostia. And then there's this little space. And that space is called the incurrent canal. Right there. And from the incurrent canal, it comes out another opening right here. That opening, that's called the prosopile. And the prosopile is an opening that allows the water to flow into the radial canal, which is right here. This is the radial canal. I'm going to try to like zoom in when I edit this to see if I can do that. That'll be a new skill for me. Probably take me a month to do it. But anyway, that's the radio canal. And then it comes out this hole right here. That's called the apopyle. And then as it comes out the apopyle, it comes into, once again, the spongiocele. And then from the spongiocele, it goes out the osculum at the top. Okay, so once again, this is a body type, a body plan, or a body style. This is not a taxa or a taxonomy. We use these characteristics to classify them later, but what we're talking about here is like a body type. Kind of like if you had maybe people that were really short versus people that were really tall. Maybe that's not a good example, but they're all the same kind of animal, but they have different. They come in different sort of styles. And in case like, I think the problem with height is there's this range of variation. So if you just had short people and really tall people, it'd be more like that. Maybe it's a bad example. Okay, but anyway, that's what you have there. Then our third body type is called the leuconoid body type, and it is the most complex, and it has ostia, incurrent canals, and then it has these flagellated chambers on the inside, excurrent canal, and the osculum. So these are the flagellated chambers that you can see right here. Now I will tell you that in terms of labeling the parts, this gets way more confusing because the water's flowing through a very non-uniform shape often. So the ostia still makes sense because there's holes there. The flagellated chambers still make sense, as you can see that's where the flagellated chambers are at. And the osculum, when it comes out somewhere, kind of makes sense, but there can be more than one. But it's far harder to label, like, what do you call this part or that part? So it's not as clearly defined on that. And so I wouldn't ask you something in the middle of nowhere here, like, what are we going to call that? Okay. Now, that being said, we then have our classification system. How do we classify them and name them? And so remember now, we have, just for the sake of keeping things consistent, supergroup, we're in the Uniconta, all right? And then the kingdom below that is Animalia, right? And then we have the phylum Porifera. We talked about that. So now we're at class. So domain, kingdom, phylum, class, and we're going to keep working our way down as we do this. So it's this nested hierarchy of more or less complexity depending on the direction you're going. So class Calcarea then are sponges that have calcium spicules, and they come in all three body types: asconoid, cinchonoid, and leuconoid. And so what that means is there are many different species of them, and some of the species are asconoid, some are cinchonoid, and some are leuconoid body types. Depends on the species. Then we have the Hexactinillidae, and we have a really nice one of these in lab, and these have silica spicules, and they come in only two types, the cyclonoid type and the leuconoid type, and they have this very glass, it's like a glass-like material that acts as this three-dimensional skeleton of the sponge, and you can clearly see that. You can pick it up and you can hold it. Then the last group is the Demospongia, and the Demospongia have silica spicules and or spongin, and they only come in the leuconoid type. This animal here is a sponge, is a Demospongia, mostly made out of this spongin material, but it also has the cells to move the water around and that kind of thing as well. And what's interesting, when you go to, well, let's say a long time ago I'm guessing, this is a guess, and guesses sometimes aren't good, but let's say the 1800s or something, in the long time ago. Anyway, original sponges were actually this animal. You know, that's, you know, and you can, I don't know if you can still do it, but occasionally you can go to places that sell, you know, things that you would put up on a shelf that would be interesting maybe, and you can buy these sponges. They're not the sponges we use now, like to clean your kitchen. We make those out of cellulose, but that's based on this same kind of porous model of sponge that was really this animal. And every now and then, this is sort of the classic real life sponge, and that's where it gets its name from. And we turned that into something later on without using the animal. But anyway, there you go. Okay, the next group of things we're going to talk about have radial symmetry, and they include the phylum Cnidaria and the phylum Ctenophora. So there'll be two different phylums here. These will include that things that you're familiar with, like sea jellies, which you used to call jellyfish and maybe still do. You know, like I told you, this is where you get new groups of friends, because the fish part, the jellyfish part, I mean, and it's simple and subtle enough, and you think it's okay, doesn't really matter, and maybe it doesn't. But the problem is then when you get to talking about fish or that kind of thing, people then think that thing's a fish because it's got it in the name. And so anyway, sea jellies is a better term for it. Sea anemones, Finding Nemo, if you watched that or go back and watch it, sea anemones are in that group. Cone jellies you're not as familiar with, but I'll show you those. And they have a tissue system level of organization, what we're doing now. They are diploblastic. Diplo means they have two layers of tissue. Those are the endoderm and those are the ectoderm. So you might remember when we talked about the developmental model, and I said, you know, we're doing this kind of thing. We're taking, it looks like a mushroom, but it's like a ball, you know, and you're pushing your thumbs up into the ball. When we talked about the gastrula, right, as we were forming that stage. In these animals, this kind of development happens, but there's only two tissues that result, and they are endoderm on the endoderm on the inside and ectoderm on the outside. So as this pushes through, you end up sort of with that kind of thing. And this is the endoderm tissue. And the outside, you have a different tissue. You have ectoderm. Ectoderms ends up forming the skin and things of that sort. And the endoderm, the digestive tract, which is very simplistic in these organisms. So just like we have with sponges, they come in two different body forms that are very distinct. One's called the polyp stage and one's called the medusa stage. But unlike the sponges, many of the cnidarians the same animal will be a polyp or a medusa at different parts of its life cycle. So there are some cnidarians that are always polyp, and then there are some that are always medusa. But then there are some cnidarians we'll talk about where the body type changes depending on its reproductive cycle and things like that. So this is my typical polyp type, and this is my typical medusa type. So when you think about a polyp, if you think about Finding Nemo or a sea anemone, that's your basic model of a polyp. And then if you think about the medusa, then your sea jelly, you know, your jellyfish that we don't say anymore, but your sea jelly, that's the same as sort of the medusa stage. And if you kind of look at these two, if you took this one and flipped it over, and then sort of squished it, you kind of would have this form. So you can kind of see how you could have an organism shift between those two types. Okay. The other thing that you find in Cnidarians that they're well known for. And this is one of the nice things to think about Bio 2 when we get to this part. We get to animals, and again, although there's a lot of information to cover and it can be overwhelming, the nice thing I think about where we're at in Bio 2 now is we start getting to things that you're familiar with, that you've known at least, you know, since you were a kid or you've heard about. So that allows your brain to sort of connect it to things you already know. And I think that although there's a lot of information, and Bio 2 gets harder at this point in terms of how thick your packet gets, I think that we find that students, as we go into this, start doing better normally. And I think part of that is because it's more familiar to them than something like Plasmodium, or, you know, it's harder to sort of understand some of those cellular, smaller things because you're not as familiar with them. You haven't seen them your whole life or heard about them. So when we talk about Cnidarians, they have these specialized cells that you'll have kind of remembered or know about. May not know this name, but they're called cnidocytes. Cnidocytes are the cells that inside the cnidocyte have this structure that can be shot out, and it's called the nematocyst. So the nematocyst is the structure inside the cnidocyte cell that can launch out this thing right here. Here's my nematocyst, and that's the stinging cell, if you will, of a sea jelly. But so everybody knows about, you know, you can get stung by a sea jelly and it hurts. And the way it hurts you is because you can get stung by this nematocyst. The cnidocyte's the cell, and the nematocyst is what shoots out and stings you. And then the trigger for that is this little feature right here. It's called the cnidocyle. It's part of the cell that triggers the release of that nematocyst. And this is actually, it's interesting because this is a complex sort of thing. These respond to different things like chemicals and touch, and there's different kinds of nematocysts. So different Cnidarians have sort of a different sting, if you will, depending on what is around them. And some of them have in that nematocyst, there are toxins on that. And so not only can it hurt you, but then the toxins can be really bad. And in the United States, you know, things are fairly safe for the most part. But there are places in Australia we'll talk about as we move on, where they have some of these Cnidarians that, like, you get stung and you die. Like the death rate is high if you get stung by it. But we'll talk about those as we move on. We have 4 different classes in our Cnidarians: Hydrozoa, Scyphozoa, Anthozoa, and Cubozoa. These are hydrozoans right here. Just a couple of pictures of them. The hydrozoans are mostly marine. There's a couple freshwater ones, and we'll look at one, or we'll have one in lab in the genus called Hydra. And they have both a polyp and a medusa stage. And when they're in the polyp stage, it's often colonial, like they live in a big colony of them. And they can reproduce 2 ways: asexually by budding, kind of like what we saw in a paramecium, sort of, not quite, but kind of. And they can reproduce sexually, where they have sperm and eggs and they form zygotes, like you see in this picture here. So this is my polyp stage right there. Then this is a reproductive polyp that then produces medusa, and then the medusa can go out and reproduce sexually, making sperm or eggs, and travel, and then that can implant later on and make a new polyp stage. Scyphozoans are more like our sea jelly that you're used to, and they are all marine. Polyp stage is reduced or absent, and the medusa stage of it is free living, and we commonly call these, like I said, sea jellies. And then our last group of these is the anthozoans, and the anthozoans are all marine. They have polyp stage dominant, no medusa stage. So things like our sea anemone, again, Finding Nemo, That might be like, you're like, what's Finding Nemo? It's unfortunate. There's the, as a teacher, when you start, you have these stories and things, and Finding Nemo's like way before my time. It's a classic, so hopefully we went back and watched it. But one of ours is Run, Forrest, Run. We used to say that, and students now are like, what are you talking about? We made our kids watch it the other day so we could communicate with them still. But anyway, that's a sea anemone, and we'll... on field trips, if you go down the tide pools, you can see them. The last group then are the Cubozoans. These are the ones I was telling you about in Australia. They're also called box jellies, and they have these complex eyes on the top of the head of the Medusa stage. There's one in particular called the sea wasp that's very, very dangerous, and the venom from that, there's enough venom in one of those organisms to kill 60 people if you get stung by it. And many years ago, we went to Australia, my wife and I, and down by the beach, when the box jellies are, all over and reproducing, and there's a bunch of them in congregating, or they're around, they put up signs in Australia, they put up signs saying, don't go in the water. And guess what you see, which kind of blows my mind? You don't see people in the water. Because in Australia, when they say, hey, the sea wasps are out, don't go in the water, the Australian people listen, because otherwise you end up getting stung, and that's the end of you. So it's kind of amazing to me, because here, when we put signs up on our beach saying, hey, don't go in the water, you still got people, surfers, going in the water. It's a very different kind of reaction to that. So this is in the wrong spot, but here's my other pictures of class Anthozoa, and I always forget to move that up, just another picture of them. Okay, I'm going to stop there, and I'm going to do the ctenophores next time. Thank you very much. Hope everyone's having a good day, and I'll see you soon.